Oh, my wife has 500,000. Why would you open those stupid emails that you get every time you sign up for, say, Cole sent you something and whoever? I mean, I could care less. If the email, every time you guys send me, you email me and come back, oh, thanks for taking care of that. I can see it right away. Why would I have to bother opening it up? It's annoying. I know what some emails are. I can search. Oh, everybody comments on that. You too, Chris.
Yeah, I did that today.
You're terrible, all of you. Nobody likes anybody. All right, we're going to finish this chapter, I hope, today. The last part we did was the HIV thing. Really, for me, for this chapter, the parts that are important, how it's classified, how it spreads, and if there's a certain thing that I emphasize, and when I do the review reporting, certainly, like rubella being involved in crossing the placental barrier and causing problems, typically nervous system and hearing, or HIV being a unique because it's a retrovirus. We don't get into all the other stuff just yet, because we revisit a lot. We revisit nearly all the ones I talk about in subsequent chapters. This is where your class is going to probably diverge from Dr. Dress's and Mrs. McGrand's chapters. Is I just call it. When we begin to diverge from that, because my thrust is much more clinical. So I like to talk a lot about, I'm sure everybody talks about antibiotics. I talk about diseases a lot and give you some background. I think that's a better way to understand. That's just me. And everybody has their own way of doing it. You get all the stuff that you need. But the next chapter we're going to talk about here is methods of control. And we started to talk about it. That's a little bit of what you did in lab when you did the temperature lab and you started heating up stuff. I'm sure everybody did, right? Whether you're my lab or not. So that's kind of where we're going with this. So I'll go over all those things, and there's not a lot left in this chapter. There's a couple of things I do want to emphasize today, and then maybe introduce you to the methods of control. This is not, when we get to it, it does not involve antibiotics. We come to that later. So voila, here we go with all this stuff. So this is the PowerPoint. This is the continuation. I mean, I just flipped through all the pages. If you saw me before class, we were here, and I just ignored all this stuff because with phage bacteria, really not that relevant to you, whether it's temperate phage or lytic phage. But when we come to this, it's something called lysogenic conversion. A lysogen, okay, means, if you recall, phage or bacteriophage viruses, they don't infect us directly. They infect bacteria. The trouble is, very often, though, just like DNA viruses do with us, like a herpes virus would do with us, they incorporate into the genetic material of the bacteria, into its DNA. And so at that point, a couple of things. When they call superinfection, means once it's done this, it kind of hardens the exterior so it can't get another virus to reverse it, so to speak, or to damage it. So we call that a lysogen. And what happens, it can, because of the genetic material of the virus, can alter the DNA. We call that lysogenic conversion. And just to give you an idea of what a role this plays, botulism, the toxin from botulism comes from a virus like that. The toxin from diphtheria, which we'll get to in respiratory diseases. That really the most dangerous and maybe the most notorious strain of E. coli, O157H7, this toxin. Salmonella, food poisoning, which is really better stated, it's called food infection. Down the road, we'll get to the distinction between those. Again, an alteration. Strep pyogenes, we're going to get to all the streptococcal problems. One of the worst historically before we had antibiotics was scarlet fever. Killed untold numbers of people. And cholera, one of the worst diarrheal diseases we see. And all of them have the severity is because they have been converted or altered by a virus. Make no mistake, viruses can affect every living thing, every living domain. Archaea, bacteria, like us, eukarya. They can affect plants, so we really don't deal a whole lot with that, but they're very significant agriculturally. So you should be aware of lysogenic conversion. So I mean, I wanted to make that the jumping off point because I'm just going to just hop around in this PowerPoint, go back and look at the other PowerPoint, see if I missed anything, and move on. Fair enough of me droning on about viruses for now until we get to the actual disease. Hey, I got to de-drone. So I'm not going to do filamentous and nobody. We're going to get, when we do, side note, I'm going to for this class, for a couple of reasons. I'm going to de-emphasize the genetic material. We're going to do a little bit of that. That's where this thing called transduction is. It's kind of the way that viruses can impact different things. And bacteria are a good example of this. We'll take a look at this, what transduction does down the road. But for now, just leave it, whether it's specialized or generalized, because it's just going to be a little bit confusing. Okay, because this is sort of has to do with the inherent genetic instability of bacteria. Like I said, the virus can introduce a toxin. Just have you, interestingly enough, that toxin actually has the ability to spread to other viruses. It's sort of how MRSA, how a population of Staph aureus can become MRSA overnight. We're going to get into that. And transduction is one of those things. We don't do these plaque assays in your lab. In the advanced labs, when we're studying genetics and biochemistry, the advanced, bio pre-med major labs, this is where they study those viruses and these plaques. This is kind of where effectively what they're doing when the bacteria phase is there, they're killing the bacteria. That's why we're looking at them possibly as an alternative to antibiotics, because as you're going to see, part of not this unit. When we start talking about the next test, it's going to be a lot about you're in such big trouble because we don't have enough antibiotics that work anymore. This is important. This is important not only for me, it's important for you going forward. This is the kind of thing that NCLEX and licensure exams, they might talk about. It's how viruses, and again, we classify them as animal viruses because they affect us, part of the animal kingdom. They can't hurt you until they attach. So it has to be receptor based. Again, if you have something that affects you respiratorily and it gets on your skin, it's not going to affect your skin. Okay, but the trouble is touching your face, do something with your hands. You might do that. The next thing you know, you've sort of aerosolized it. You can inhale it. So the fact that it's on you, it makes you at greater risk. That's why we're going to keep going over the same thing: hand washing, hand washing, hand washing, hand washing. So first, it has to attach to specific receptors. HIV is such an oddball. I really don't do it in this class. It actually requires two. It's got a receptor and something called a co-receptor. It's very interesting. Okay, but take it from me. First, it has to attach. And again, the range varies. So measles, us. So when I had the measles as a kid, we didn't have to worry about it. We didn't have a dog, but my kids didn't have to worry about it because they got immunized. But what I'm saying is, they don't get that particular disease. Rabies, very, very broad range. This is the interesting part. Enveloped viruses or non-enveloped viruses. So enveloped or naked viruses, it doesn't matter which it is. Once they attach, they are brought into the cell. You don't have to go into the distinction. Either the membranes fuse or it's sort of endocytosed. Just the way we remember endocytosis from AP1. Basically swallow it up, the membrane kind of surrounds and hugs it, brings it in. The big step, all of this is very, very similar to the way the phage viruses infect bacteria. The big step is phage viruses just inject, genetic material. We, in animal viruses, bring in the whole thing, the nucleocapsid. So you have the capsid, the protein shell, and the reason I'm making a big deal out of this was the first kind of antivirals that were produced. Because what happens next is this process called uncoating. So uncoating means we have enzymes in our cells, or animal cells, to basically remove that protein coat and allow viruses' genetic material to escape and then effectively takes over. So uncoding is a very important step. The earliest antivirals were designed for influenza. They were anti-encoding agents. Reality is we don't use them because they just weren't very effective unless you got them even before you knew you had the flu. And that's why there's other better, Relenza and other ones that work very, much more effectively get on top of the flu early on. Just saying. So we'll get to that when we do the antivirals. But that's the big step, uncoding. At that point, it takes over. Okay, what it does, it basically hijacks all the host cell's machinery for transcription and translation. And then at which point, if it's an RNA virus, it makes more RNA. Then it makes proteins. It makes proteins for the capsid. It makes protein spikes. Like HIV, it'll make proteins that go inside. Doesn't matter. So it basically takes over and becomes effectively a little factory. It does not replicate the same way. Our cells replicate, we know, by mitosis. Bacterial cells just divide in two. It's called binary fission. Viruses don't work like that. Effectively, it's like a little virus assembly line. We're going to make more genetic material, more capsid proteins, more protein spikes, any other proteins that go with it. And it's like a little assembly line. We're going to make it. We're going to put all the pieces together. And it's either going to reach a thing called a burst size, when there's like 1,000 inside one of our cells, and it bursts. And then it can affect another 1,000 cells. And those 1,000 cells can infect another 1,000 cells. 1,000 times 1,000, that would be 1,000,000 cells. That's why viruses work very quickly. If you have a large burst size and they replicate rather quickly. Other viruses, particularly the enveloped ones, they become little factories, but the cell will never be the same. Your cell will never get over the virus. It's going to die eventually from it. But really just starts spitting. You'll see some illustrations. It just starts spitting out viral particles. We call it budding or shedding. Typically, those are enveloped viruses. Naked viruses typically burst, like the common cold. Coronaviruses, for instance, would bud or shed because they're enveloped. That's how they work. And now all of this is something I don't go into with positive and negative sense strands for two reasons. One, it confuses the heck out of me. I can only imagine how confusing it would be to everybody else. But for like our students in, advanced genetics and advanced biochemistry, this is stuff that they need to know for their careers going forward. I don't think you do. Okay, and I don't think anybody does. So we call it the transcription strategy. It's kind of how they replicate. Don't need it. Okay, all of these things don't need it. Don't need it. Don't need it. The only one that's interesting, and I think more than anything else, if they have it here, and I'm looking for, the one that I commented on before, because basically it's the unique factor that has to go with HIV. HIV starts as an RNA, and remember, then it sort of comes into the cell, and that enzyme reverses. So it's complicated. That enzyme that HIV has called RNA reverse transcriptase rewrites that RNA as DNA. Remember, that's the area where we see mutations, why we have to use multiple antivirals here. And then the DNA expresses itself as RNA, go figure. RNA backwards to DNA. Now it takes over, it makes more RNA, that makes more proteins. So there's a certain amount of complexity to this, and then finally it makes viral particles, and effectively they bud or shed. So down the road, there'll be more about this, particularly when we look at the antivirals that are involved. So, and it's rather, and there's more steps than that, but this is just sort of the basis that's going on with it. And again, I don't want to go crazy with it. Why COVID? Why influenza? Why respiratory syncytial virus are such a pain is they do this. They mutate. Commented on it briefly before. Remember I talked about antigenic drift and shift. Drift are subtle variations. It isn't necessarily going to make the virus terribly worse. It's not going to make that flu worse than it is. The trouble is if we make vaccines ahead of time. I mean, maybe you're wondering yourself why we have how many variations of COVID. It keeps mutating. And so the vaccine they made for Delta isn't going to work for Echo or Mu, whatever Greek letter nickname they're going to give it. It just doesn't work. Oh, it has some activity. Yeah, it has some activity. It doesn't have total activity. The problem is when it mutates, it makes whatever vaccination or immunization, whatever you want to call it, less effective and sometimes ineffective completely. That's why seasonal flu shots don't work. Last time I got a flu shot, it worked because I got the flu in April. I remember my family doctor going, "Hey, I gave you a flu shot in November." I said, "You did?" Because you got the flu. I said, "Bingo." And I looked at him and said, "That's the last time we're having one." Not helping me.
The same thing happened to me every time I get a flu.
Don't bother.
I know.
The trouble is if you're going to understand Stein's opinion. Okay. My wife hated it. My wife worked for UPMC.
That's what they required.
Required.
I had to get it.
You had to get it. So my wife worked, and she was upset about it every year. I can remember she started as a nurse's aide, and then she became an LPN. And I mean, for the dozen or so years she worked that way because she didn't start that career until she was in her 40s. Okay, and it bugged her. She's very anti-vax. I mean, I'm not, it's okay. Everybody's entitled. I mean, I'm a little more moderate about a lot of things, but she is not. But that's okay. We only get into limited arguments. But once again, you know where she dragged me last night? To the gym. She comes back from running around, pulls the car, and she starts putting on her sneakers. I said, "We're going to the gym." She goes, "You bet we are." And again, I knew exactly what to say to her. It's all right. I was doing good. This is the problem. The antigenic drift are these tiny variations. And we'll go over this again, when two different viruses or strains infect a host, you can get a strand from one and a strand from the other. And that's how we saw pig DNA or bat DNA or some type of bird DNA get into human DNA, or rather RNA, I beg your pardon, get into an exchange place. That made that so-called novel virus. We will revisit this when we talk about respiratory diseases at length, just to acquaint you. And we're not done yet. So here's that reverse transcriptase. I could save myself the trouble writing it, but you can see what's going on. This is a very complicated passage. So I don't expect you to read this or look at it. If you're interested, fine. Yes, ma'am.
What's the difference?
Okay. Shift suggests that a whole chunk of RNA, remember there's multiple strands, a whole strand from a different species got incorporated. That's how something that would affect us all of a sudden has a whole strand of something from a bird. We call them avian. Or from a pig, we call them swine flu. And COVID was a little bit different because it seemed to have elements from multiple species. And again, I'll send you the textbook's PowerPoint about it. Again, science. They're playing the company along. Meaning, they're a textbook for McGraw-Hill's giant publisher. Giant. And I mean, they have to put in their books that the majority of people are going to want to see them. And offend is the wrong term. It bothers me when they tell you how many millions of people died because of COVID. Anybody, 100-year-old person dying in a hospital who happened to test positive for COVID, oh, they died because of COVID. No, they died because they're 100 years old. maybe that was the last push. Oh, maybe, but they weren't going to last 110. Didn't significantly shorten their lifespans. When you turn around and put everybody who had COVID, elderly people or infirm people into the same nursing home, what the did you expect to happen? They were all going to get better? No. You were basically keeping, want to look at it that way, they were trying to keep the infection away from the healthy. I'm sorry. Go ahead. Somebody tell them that's, so you're intelligent people. You're young, intelligent, bright people who are only going to learn more with time. Read what's out there, form your own opinions, please. That's what's going on. So to answer your question, I'm sorry to get off on that tangent, but the reality is that's what makes the shift so scary. We have been worried about that since they finally recognized that the worst infectious disease of modern times, that Spanish flu of 1918, 1919 that killed so many people, we've been panicking about that. They had it. If you didn't know, back in 1976, I just got a suspicion none of you were born then. None of my kids were born then. Only a handful of my colleagues were born. We were sitting there and they had a swine flu outbreak, they thought, and they formed a vaccine relatively hastily. And they started to immunize. They were going to start, this was under President Ford, President Nixon had resigned, going back to your history. And all of a sudden we're hearing about that. And they stopped the campaign because they developed an unusual side effect to immunizations. It can happen anything. It can happen with immunization, it can happen if you have a viral condition, it can happen without anything really to predict it. It's called Biol syndrome. All of a sudden, people were getting the first cohort of people receiving this vaccine up in Minnesota or Wisconsin. They had about a half dozen cases. It's an ascending paralysis. We see post-viral infections, post sometimes immunizations, post sometimes, there's drops in people's immune system. And all of a sudden, it's like the reverse of polio. You begin to, as polio kind of starts and goes this way, ascending means if it starts in the extremities, works its way up, and once it gets to respiratory, it's like that. And all of a sudden, this vaccine that they were going to give out nationwide, stop. They're afraid. It turned out statistically, when they did the numbers, it wasn't any use behind it. So, I mean, a lot of times we have had an overly exuberant response historically to perceived, world life-threatening diseases. Yes.
That's ALS, right?
No.
ALS. Oh, they sound really similar.
They do sound Guillain-Barré syndrome. They do. Or ALS, okay, Lou Gehrig's disease, amyotrophic lateral sclerosis, impacts both the nerves going from your brain to your spinal cord, and from your spinal or cranial nerves going out. So it's a little bit different. This paralysis kind of came this way. So I mean, the result is paralysis, but there's significant differences between the two. I can't wait. Alex.
Sorry, I know we're a little bit past.
Oh, viral butter shed. I know. I'm the world's just go. That's why I type stuff for you. I'm a terrible typist, but I type a lot. I was spoiled. My mom's a professional secretary, and she would not. She was so smart. She'd proofread. I had great papers because she'd proofread them and do all that type. Back in the day, we didn't have spell check and things like that, or all the type of things we can do with Word, stuff like that. My mom was great, and so I learned to be a good typist. But she was like, those are the days you could take shorthand as fast as you could speak, and she could type 125 words a minute, sustained copy. Like she'd get listening or watching shorthand. Never looked like that. I always wonder what happened if my mom and dad had had the luxury of going to college, what they would have been like. Boy. And that's it. Assembly is when they put all those pieces together. And then finally, most of the enveloped viruses, a few do burst, typically leave by something called budding. And they basically, what happens is the protein spikes get embedded in the membrane, and they pick up a piece of membrane as they're leaving. The non-enveloped or naked virus is typically the host cell dies. When they talk about bursting, basically they're inducing that host cell to undergo apoptosis, and all of a sudden, and this is what. So this is actually what it looks like micrographically of budding or shedding. Here, the little viruses are in there, and all of a sudden, kind of on their way out the door, they pick up a little bit of membrane. The spikes are in there, and they're showing it to you. It's almost look like they're being. It's not exocytosis because there the membrane opens. Here, they're sort of getting the membrane. The spikes have been implanted there. The membrane folds around them, and voila, they're released. As we call budding or shedding. Okay, and the difference. Okay, this is sort of generic about infections 101. Things that they do. They either bud or shed. Okay, I beg your pardon. I was looking at something. They either are acute, the cold. Okay, what happens with a cold? It comes on fast and it goes away fast. Most time the colds, they're really nasty. A couple of days to get it, a couple of days to have it bad, and a couple more days to get rid of it. Over a 7 to 10 day period. Okay, and that's all it is. When they're released, eventually your immune system does the job. See you later. There goes the cold. Persistent is the term we use here. Some people will say chronic instead of persistent. Okay. And a lot of times, persistent infections, people are asymptomatic. We see this with viruses. We see this with other problems. Okay. Sometimes they're even bacterial. Very famously, Typhoid Mary was shedding, but not viruses. She had bacteria. She had a salmonella bacteria. And she never was sick, but she was handling food for very wealthy people in New York at the turn of the last century, 1890s to 1900. And because she was a very famous cook. And so they all got infected and they were dying. And she wouldn't believe it. Well, I've never been sick a day in my life. She was what we call an asymptomatic carrier. You can have that for viruses as well. Okay. And again, now this is interesting because some viruses have an acute phase and a persistent phase, and then they're talking about HIV. You'll see that in a second. So a lot of times we talk about chronic infections. And here are the asymptomatic carriers. And then the difference is a latent infection is like what happens when you get a cold sore. You get a cold sore, it's there. You may never get another one again. When you get a wart, it's there, and eventually it goes away. You may never get another one again, but you may. That's what latency is. Why you get it, not clear. Some fluctuation in your immune system. Again, there's a lot of variation. A lot of times when we look at this and we look at that skin diseases, a lot of things cause your immune system to weaken. And it doesn't always have to be physical. I mean, again, emotional, social, loss of a loved one. Would I be surprised if my wife who used to get cold sores worrying about nursing exams, would have had one develop because her mom died? No, because it in some way, shape, or form, that's the reality. We don't understand all of these things. These are examples of ones that persist. The herpes, particularly, it becomes latent. And down the road, and you can see where it is. It's in the salivary gland. It'd be in your white blood cells. The same thing, Epstein-Barr. This is a B cell is a white blood cell. It's a lymphocyte. Which are involved in antibody production. And down the road, okay, because you could get mononucleosis, which could recur. There's a rare cancer we don't see so much that affects salivary glands called Burkitt's lymphoma. Okay, that's there. Cytomegalo, which is technically in the herpes family, these are really immunosuppressed individuals. It's unusual for healthy individuals to have it. Hepatitis B and hepatitis C, the problem with them is because liver cells replicate so rapidly, they unfortunately can undergo significant damage in that line and develop particularly carcinomas, deadly changes in the liver. They can induce cirrhosis, chronic liver disease, even though by far and away, I think we see it this way. Alcoholism is the predominant cause of these problems. My feeling is that the alcohol, you're weakening your system so much, you're really predisposing yourself to get these viruses if you come into contact with them. So it's sort of the chicken and egg thing. So I mean, it's tragic, but it's true. So here, the most common, herpes simplex, HHV1 and 2, classically latent. Where do they hang out? Sensory ganglia, any sensory nerve. Okay, so typically oral, and we see a lot of recurrent with this. We even see it recurrent genitally. The genital ones are certainly more problematic and painful. And when we revisit this again, we talk about viral diseases sexually transmitted, depending on what type of sexual intercourse that one chooses to have with a partner. Oral can be genital and genital can be oral. And then HIV famously affects helper T cells, and helper T cells are so important. They're the linchpin of your immune system, and we get AIDS. And we'll get to this for those that unfortunately have me for A and P. We're stuck. And then lastly, the one that we've all effectively, that's varicella zoster. Again, notice the herpes viruses predominate here, chickenpox, and then shingles. And it's a good, I mean, it gives you an idea. This is a typical pattern. This is sort of, I'm getting sick, the exposure, the time that it's growing, incubating, if you want to call it that. The actual, I mean, this would be influenza typically. Maybe this poor phase would last 5-7 days and get better. What happens with the typical herpes virus, it's there, it goes away, and you're non-infectious at that point in time until something reactivates it. It could be months or years. Okay. I mean, and the same thing, if this were shingles, the same would be true. A child who had the chicken pox or a child who was immunized, anytime after 6 months, theoretically could get the shingles. It's the same virus, it just presents differently than the initial phase. And then hepatitis B or hepatitis C, this is the problem. When it becomes chronic and persistent, it's always in your secretions. That's when you have to worry, particularly vis-a-vis hepatitis C, or even in AIDS would be the same thing. AIDS is kind of like a hybrid of the two of these. It's not there, but it progressively it kind of does this. So what happened, if it's untreated with HIV positive, you have HIV, which is a mild infection. There's nothing memorable about it. It's like having a cold. If it goes untreated or people don't recognize it, okay, or there's a lot, and we'll get into that more when we get into the disease itself. What happens over time, it kind of goes from this, it enjoys a certain degree of latency, and then gradually those particles reach a point where they are constantly in your system, and at that point your immune system, because it really signals the collapse of your immune system. So you on the firing line, if someone is HIV positive, you have a comparatively limited risk from an exposure, a blood splatter, a needle stick, maybe 1 in 500. When you get to AIDS or chronic hepatitis C that are being untreated, then you have a very significant risk because it's in all of their secretions. The moral of the story is when this happens to you, like it happened with my first wife, even though she was running that nursing home, you report that to your supervisor. That's why the good Lord made incident reports. Don't let somebody tell you because it's going to make them look bad. Oh, don't worry, that was pretty minor. The only thing that protects you and gives you an opening into workman's comp or having a disability leave associated with this is filling out the incident report. Do that. And if it's your subordinates, make them do that. Because there's an old expression we use in life and in medicine and in the hospitals called CYA. It's the only thing that covers your A. I was going to say yes. Oops. So CYA at all times, because if you don't, ain't nobody else going to be doing it for you. Please. Don't let somebody, and the only appropriate term is bully you, because you're a newbie. We really don't have to report that. I'm Phil. You have a choice. You can either give me the incident report or I will go to someone else and make sure I get it filled out. They may not love you, but heaven forbid you end up with an exposure like my ex did, now all of a sudden you have something you can hold on to that protects you going forward. So here it is. Those are sensory root ganglia. That would be the trigeminal nerve you're looking at, etc., and reactivating it. The other thing I think is really important. Hopefully, we probably won't get through all of this. We'll end up having to do prions probably at the very end. Tumors. So the short course. These are the viruses that directly link to cancers. Okay. Hepatitis B and C because of the nature of those, the rapid reproduction, because they are not in a typical case. Kaposi sarcoma, herpes virus, Epstein-Barr, Burkitt's lymphoma, HPV, lots of different ones, certainly cervical carcinoma. And this is an odd one with a retrovirus, okay, that adult, so it's relatively rare. I don't really go into it as much. Here's how they do it, okay? Effectively, beautifully stated. In your body, you have genes that when they are activated cause tumors. They affect that cell cycle that we talked about in AP. They're called oncogenes. Normally, they are inactive. The inactive stage is called proto-oncogene. So they are inactive when they're versus the active form called an oncogene. Certain viral infections can activate. The other ones that are always active are called tumor suppressors. They are always active. They can be inactivated by certain viruses. So the best analogy, it's a wonderful book that won a Pulitzer Prize back in 2010. I listened to Butler. Basically, he talked about, it's called The Emperor. He almost made a biography, a life biography for years. Cancer, called The Emperor of Pulmonology. Looked it up on iBooks or Kindle, you'll find it. Good. Very readable. You're at the point now you can start to be able to read that. Because you're starting to get the background. So, I mean, it's not an easy read, but it's interesting. So, effectively, what this is his analogy, not mine. I'm not that smart. This is the accelerator, these are the brakes. When you hear about the breast cancer genes, what the breast cancer genes do is they deactivate or they impact those tumor suppressor genes. So what happens with certain viruses, what happens with certain cancers, they will turn proto-oncogenes into oncogenes. That's like your accelerator in the car getting stuck. Well, my accelerator's stuck, but at least I got my brakes. You see it, Sydney? I know my handwriting is good. They go from proto-oncogenes to oncogenes. It's like the accelerator in your car. Does that work? Hit him. Tumor suppressor genes are like the brakes. Well, my accelerator's in trouble, but at least I got my brakes. When tumor suppressor genes that are normally activated, like the breast cancer genes, are deactivated, I lost my. Imagine if you're in a car with the accelerator stuck and your brakes are going. You're going to crash into something. You're going to start. It's like a bad action movie. All of a sudden, oh my God. Somehow, the superhero escapes. So, and all the things we'll talk about cancers. So, proto-oncogenes stimulate cell growth and division. Tumor suppressor genes inhibit it. So, mutations in these or invasion by viruses, multiple changes. The oncogene is the proto-oncogene that has been changed to promote uncontrolled growth, not normal growth. So, a virus can disrupt it. Chronic infection or inflammation can do it. A virus may even carry these. HPV is associated with cancer. So that's the idea. proto-oncogenes and proto-oncogenes, etc. I should type something up that makes that clearer for everybody. If I'm feeling spunky today, I will do that. Because I'm giving exams today to my other classes, not yours. Yeah, ignore that. Yeah, we grow viruses. How do we cultivate viruses? We cultivate viruses in in tissue culture. So, I mean, that's where when you get immunizations, typically for viruses, they grow them with bird eggs. So if you have an allergy to poultry, let people know. That is a problem. So it's typically chicken eggs or duck eggs that they grow them in. And then these are some of the bad things they do because, see, basically when you're infected by a virus, its impact is through your immune system. Sometimes they will cause danger. They call it they'll damage cells, cytopathic effects. So syncytium, fusing the cells, respiratory syncytial virus. A lot of times we identify them by the remnants of their genetic material. We call those inclusion bodies. So that's how we identify them microscopically with stains. That's how when I was sent out a piece of tissue that I thought was a wart, okay, it would come back positive, inclusion stains positive in the stratum granulosum, and that was proof positive. That's what it was. There's different ways you can assay them. We're not going to go into that. We're not going to quantify it, but these are different things that they can do. And we're not doing plant viruses or viroids, which are RNA, because they really don't impact us. We'll get the prions in. It's the very last thing. Prions stand for protein infectious agents. All they are, and why we look at them here, are proteins that are misfolded. And what we think they do, emphasize the word think, they bump into other proteins and cause them to misfold. So they induce these folding irregularities that are there. They are linked to slow. Slow, by definition, means 2 to 10 years. It's kind of like HIV becoming AIDS. Fatal human diseases. And we don't know a lot about them. And yes, they're usually transmitted within species, but mad-cow disease ended up killing 170 people in England. Not to mention how many thousands or tens of thousands of cows that had to be destroyed to prevent the disease spread. We don't know a lot about it. We only know certain things. Know if you get it. As far as we know at this point, 1, it's slow, 2 to 10 years. It's always, this is the bad one, fatal. It's not, as far as we know, associated with cancer. But what it is associated with is this. Spongiform encephalopathy.
Is that first one say slow to?
Slow, 2 to 10 years. Encephalopathies, which means your brain. when they would examine the brains post-mortem, and now we can do it with MRI and things like that. CT scans, it had like a spongy appearance, poking holes in your brain substance. It has certain changes that we associate with Alzheimer's disease. Causes neurofibrils to tangle. I mean, these are why we think that there, and we actually know that there are at least a certain association with a limited amount of Alzheimer's presentations with prion-based diseases. So, for all we know, these diseases could all be the same. We do know that there's one that is inherited, Creutzfeldt-Jakob disease, we call CJ disease. And I'm not going to ask you about this or some of these other ones that are there. There are some variants, and we have identified it with transmission of consumption of beef. The reality is, we think that most of these are caused by ingestion of nervous system elements. Most people don't eat brains. Most people don't eat nervous system tissue. When we consume beef or pork or deer that during deer season. Basically, you're just consuming the meat, not the nervous system. The problem was that what they would do when they would slaughter a cow, they would use basically the meat from the cow. Okay, fine. We get the hide from the cow to make leather. What do we do with the rest of it? They would grind it up and feed it back to the cows. It was called bone meal. It had nervous system elements. Now that is illegal. The fact is illegal doesn't mean people don't still do it. That's why, for instance, beef that's been from this country, Japan, they won't even consider it. Okay. Beef from Canada, to some extent, they won't consider it. So it could very well be that this could be all the same thing. One of the things that's kind of told that to us is kuru. Kuru is something that when they found preserved specimens in cannibalistic cultures that had somehow not deteriorated, they found evidence of these prion-based diseases there. So this is normal brain tissue. So we call them transmissible spongiform encephalopathy. This is brain tissue with a bunch of holes. Tissue develops whole, nerves die, the brain deteriorates, voila. And it just basically induces proteins to misfold. This is kind of what it looks like. Let me take a look down here. That's the most I can tell you about it. We'll stop it there. This concludes the viral chapter, unless I think of something else, and we'll start methods of control on Friday. Thank you. I knew we'd get through that with a little work.